Great. A big thank you, first of all, to the organizers for making all of this possible, the sponsors as well, of course. So the work I will be presenting is done in the Misha Ahrens' lab at the Geneva Research Campus by myself and other collaborators, and I'll point out their contributions as we go along. Now, as you know, that some of our actions are triggered instantaneously by sensory information as it comes in, but most of them take into account internal and external sources of information that have been gathered over longer periods of time. Now, we are interested in the circuit mechanisms that integrate, store, and distribute these sources of information to guide future actions. So we make use of our mother organism, the larval zebrafish. As it makes its way through its environment, it swims in bursts interspersed by pauses of different duration. Now, this burst, for instance, this swim episode, might have been triggered instantaneously by its visual system sensing an impending threat. But it might as well incorporate past measures of behavior, for instance, how much has it been swimming in the past, or various other sensory events that have happened in the recent past. So this project set out the circuit mechanisms that does this integration. So we hypothesized that there must be neurons within the brain that do this. We first set out to find them. Now, in order to do this, we use a closed-loop virtual reality system. Now, I know you've seen various iterations of this system in the past few days, but let me just point out our specific setup. So we use paralyzed animals, paralyzed at the musculature, so that we can show visual stimuli using a projector underneath. Now, their intended behavior we record with electrodes attached to the tail. Now, these recordings will then feed back into the stimulus, thereby closing the loop. Now, at the same time, which is part of what sets it apart, we use light sheet microscopy as well as genetically encoded calcium indicators throughout the brain to record neuroactivity. Now, I know Vivek likes to look at the neuroactivity of whole cell classes. I actually quite like looking at all neurons within the brain at the same time. And we can actually do this in our system. So, for example, from bottom to top, from Rossa to Calder, we can look at neuroactivity across pretty much all of the brains at the same time. So we have control over the visual stimulus, we have a record of the behavior, and we have a view of the neuroactivity that underlies it all. Now, the project is done mostly using the optomotor response. Some of you might be familiar with this, whereby forwards-moving gratings provide the illusion of backwards motion, which the fish tries to compensate for by, with some delay, swimming forwards. So this movie shows a typical experiment with the visual stimulus shown in the top left corner, and the behavior encoded by this disk that expands as the fish starts to swim, like so. So we can now mine these data to look for those neurons that do the integration. So we did this using a very simple approach, multiple linear regression. We decided to look for neurons that meet two conditions. First of all, which integrate over longer periods of time in between movements, and which respond to stimuli that makes a fish want to swim. In this case, these forwards-moving gratings. Now, using this approach, we found that only very few cells meet these two criteria. I realize this is a little difficult to see, but there's some scattered cells in the forebrain, which is up here. But more pronouncedly so, there's a nucleus, a prominent nucleus within the hindbrain. And this is actually a surprise. The hindbrain, which is analogous to the brainstem, is much more associated with motor processes, rather than, for instance, with responses to stimuli. Now, the activity of this hindbrain nucleus can be summarized as follows. When the animal swims, indicated by these tail movement traces, the activity is very low. However, once the animal stops moving, the activity gradually ramps up with a sudden uptick when the sensory stimulus starts to move again. This is followed by a rapid decline once the animal starts to move again. Let me point out a few features of this nucleus that makes it so interesting. Now, first of all, the cells in this hindbrain integrate visual motion such that two stimuli separated in time have an additive effect on the activity of the cells within this nucleus. And actually, what I'm not showing you here is that they also respond to other sensory modalities. And what's more, they keep a record of the behavioral history, some measure of it anyway. For instance, fish that swam with high vigor, in this case a cyan trace, have higher levels of subsequent activity within these cells than do those fish that swam with lower vigor. So they keep a record of the current sensory stimuli as well as the past behavior. So what might he actually be involved in? What might be the relevance of these cells? Well, a clue came from their behavioral correlate. When you look on a try-to-try basis at the activity of these neurons, and you plot them against the response time of these fish, basically, how long does it take for these fish to respond in each trial to the visual stimulus, you can see that these two are negatively correlated. So the more active these cells are, the lower the response time, the more eager the fish is to respond. So this suggested to us that these cells might be involved in setting motor readiness. So we set out to test this. If this is true, two predictions should be met. First of all, removing these cells should delay the swim onset. Now, using 2-photon laser ablation of most of the cells in the nucleus, we found this indeed to be the case. Response times go up after ablation. Now, the converse should also be true. Stimulating these neurons, in this case using 2-photon optogenetics, should advance the swim onset afterwards. And this we also found to be the case. So these neurons have a record of the current sensory environment, the past behavior, and use it to set motor readiness. Now, to delve further into the integration by this nucleus, we decided to use a novel, yet to be published voltage sensor to look at individual spiking activity. And for more details, I refer to Takashi Kawashima, who actually is here at the conference presenting a poster. He was involved in developing this sensor in collaboration with other labs at Geneva, namely the Schreiter and the Lavis lab. Now, since we see this ramping at the population level of these cells, we expected this to be reflected in the activity of individual neurons at the spiking level. However, this is not the case. Oops. Whoops. Wrong button. These cells fire tonically. So when you look at the spike detection trace for the cell, for instance, there's no increase in the frequency of firing over time. So how can we explain the discrepancy between the ramping at a population level and the tonic firing at an individual level? Well, the answer comes from looking at differences in the activation between individual cells within the nucleus. For what you find is that cells are active one after the other in a sequence. So for instance, these fast integrators here in green are active before these slow integrators. And this is consistent between trials, between fish. So even though you don't have ramping at an individual level, because you have more and more cells becoming active over time, you have ramping at a population level. Okay, so how could setting motor readiness now happen at a cellular level? Now, a clue came from our stimulation experiments. When you do this slightly differently, not stimulating them during ongoing movement, we actually found that they suppress ongoing movement. They suppress the fish's swimming. Now, what we also found is that these neurons are GABAergic. So the fact that they suppress ongoing movement that advance the onset of future swims and are inhibitory, to us suggested the cellular mechanism of rebound from hyperpolarization. Now, the reason why we think this is such a good fit is that increasing the amplitude of hyperpolarization here on the left, or increasing the duration of hyperpolarization here on the right, within the postsynaptic neurons is accompanied by earlier firing after this inhibition is relieved. And this is perfectly consistent with our data. So in order to test whether this was a plausible mechanism that explains our observations, we decided to produce a model of our system to see whether this would make sense. Now, I don't have time to go into the details of this model, but I'll highlight some of the key features. It consists of two integrated networks consisting of leaky integrated fire neurons, one for the sensory integrators and one for the premotor populations. Now, they are reciprocally connected using inhibitory connections. At the same time, both receive excitatory input from the visual pathways. Now, the key thing is that all neurons within these networks express a rebound current, which is obviously what we set out to test. Now, using this very simple architecture was actually sufficient to explain all of our observations. Now, this is a simulation of a control situation, whereby the integrated neurons ramp up their activity over time on the population level in the absence of the visual stimulus, that we're building up a premotor rebound current. Now, after the onset of the visual stimulus, with some delay, the premotor activity comes online at the expense of the integrated neurons. Now, perhaps more informatively, we could also recapitulate our experimental data. For instance, our stimulation experiments, where we stimulated these integrated neurons before the onset of the stimulus. This leads to an enhancement of the integrated neuron activity, thereby increasing premotor rebound currents. This in turn led to the early recruitment of the premotor population. And the converse was also true. We could also recapitulate our ablation experiments. We are currently testing now whether this rebound actually occurs within the fish to set motor readiness. Okay, conclusions. These integrated neurons that I introduced you to, they integrate visual motion as well as other sensory stimuli that encourage swimming of the fish. They integrate this with some measures of past swimming, some past behavior, and they use it to set motor readiness. So they suppress swimming but encourage future swimming. And this integration occurs through the sequential activation of neurons rather than ramping of individual neurons. And we suggest that this is a new way to integrate experience and pass it on to future actions, possibly through rebound from inhibition. Okay, that just leaves me to thank the many people who have made this work possible. Obviously, Misha Ahrens has been key to this project. And Young, who joined the lab about a year ago, who's been closely involved in the experiments. Ziqian who did the modeling. Nikita, who did some of the original observations and the deviation studies as well. And then just to thank the funders, obviously, HHMI and the Simons Foundation, and the sponsors also for making this all possible. Thank you. I'll take questions.
Okay, we have time for a few questions, and please stick around afterwards for a few quick announcements. Okay, I have actually the first question.
Yeah.
Do you know much about the intrinsic currents that exist in these neurons in the two populations? I mean, I understand that a first model would be good if you just include the simplest leaky integrated fire neurons, but because you're talking a lot about sort of cellular mechanisms of rebound, hyperpolarization, you know, what do you know about the intrinsic currents that exist and potential timescales that they could bring into the
No, this is luckily for us, this is virgin territory.
Yeah.
So I could obviously say we don't know, but also we have so much yet to explore. So this is a hypothetical approach to see in which way we could be thinking, and then to test it afterwards. So we don't know about the intrinsic currents for these.
Would
things.
it be in principle possible to
Yes.
patch these neurons and basically
Yeah.
record something?
They are in a position that the camera cord film, they are very, very small. They're probably only, I don't know whether there are any electrophysiologists in the room, they're probably only 5 microns in diameter. So they are very small, but they are patchable, which is on the schedule of things to do.
Mm-hmm. Thanks.
Thank you. Beautiful talk. Do you have any idea of what might be their analogous or homologous region in a mouse, for example?
No, so these cells are, or most of them are defined by the expression of a transcription factor called GSH1. There are analogous structures in the brainstem that do also express this transcription factor, but it doesn't quite seem to correlate well with where we'd expect to find them. So basically, I don't have a clear answer of what could be analogous in the say the mouse's brainstem. No, unfortunately, not yet at least.
Okay, let's thank the speaker and every other speaker in this session.